in particular the chloroplast with its chlorophyll pigment. Okay, but these are our chloroplasts here. We're going to talk about the structures, both of the mitochondria and the chloroplast, much more in depth when we do photosynthesis and respiration. But again, similar to the mitochondria, you can see there's a bunch of folds there in the chloroplast to help increase the surface area for these enzymatic reactions to happen. The leucoplast, okay, our other example here is a leucoplast. The leucoplast is going to store starch. Okay, so remember, starch is the storage form of glucose in plants. All right, now we've got our ribosome. Hopefully you guys remember what a ribosome does. Remember, ribosome does protein synthesis. Okay, ribosomes are going to be made of rRNA and proteins, and they will help take that messenger RNA, and they will turn that messenger RNA into the amino acid chain, into the proteins. They are in charge of the primary structure of the protein. The ribosome has two subunits, a large and a small, that are separate from one another until the messenger RNA enters the cytoplasm, and then those two will join together around the messenger RNA. So we didn't really talk much about cytoskeleton your freshman year. So this will be some new stuff for you here. Okay, so cytoskeleton, some of its jobs are going to be to do cell movement. Okay, the cytoskeleton will also help move organelles inside of the cell. So organelles may need to move from one area to another, and the cytoskeleton will help that. Okay, it's going to offer support and structure, just like a normal skeleton would. Okay, and then it also can help with something that's called cytoplasmic streaming, and we'll look at what that is in just a few minutes. But these are all varieties of functions that the cytoskeleton can have. Cytoskeleton is going to be made up of three basic structures: microtubules, microfilaments, and intermediate filaments, and we'll look at each one of those. So starting here with our microtubules, okay, so we've got a microtubule here. The microtubule is going to be a hollow tube, okay, exactly like it says. You can see the picture down here. So this microtubules, they are going to play an important role in cell shape. Okay, they will also play an important role in cilia, and we'll talk in just a minute about what cilia do, versus flagellum, or flagella being our plural. Okay, they also are important for centrioles, which are important in cell division. Remember, centrioles are found only in animal cells, okay, but they are important for centrioles. And let's see what else. And they're also important in helping to move organelles. Okay, and that's one of the things centrioles can help with. So as we're looking at the microtubule here, you can see it's a tubule, it's a tube, okay, and it's got this motor protein on it that is moving this vesicle through the cell. Okay, so that's how it's going to help. Okay, in this case, it's going to help move organelles with this motor protein here. If you look here, you can see your difference between, we can kind of take a look at cilia versus flagella, as well as what a centriole is. So let's start with the centriole. If y'all remember these from mitosis discussion, remember the ones that look kind of like fireplace logs? Centrioles are going to help lay down the spindle fibers during mitosis. And you remember spindle fibers were like the roadways that the chromosomes could use when they started splitting apart in anaphase. Okay, that's part of going back to helping to move organelles as well as help move chromosomes. Okay, is what those centrioles will do when they lay down the spindle fibers. The centrioles are going to be made in what's called the centrosome. You can see that right there. Okay, so the centrosome is what's going to act is where those centrioles will be made. And again, they're only in animal cells. Over here on the right, you've got your cilia versus your flagella. Okay, 9 times out of 10. Your cilia are going to move things across a cell, while your flagella are going to move the entire cell. Now, sometimes your cilia can move the cell. Okay? Sometimes they can actually be used for cell movement, but a lot of times they'll move things across or over a stationary cell. And you'll notice they work very differently. The cilia work more like a big fan, and they all beat kind of at the same time. Where the flagellum, a lot of times the flagellum will kind of spin, okay, and that's what will propel or move the cell. In plant cells, you're not really going to have any cells that have flagellum. In animal cells, not too many have flagellum. In particular, those sperm will, so that it can get to the egg. Okay, here we have microfilaments. Remember, microfilaments are another one of the fibers that make up the cytoskeleton. So these microfilaments, as you can see, they're a little bit thinner than the microtubules were. Examples here, you've got actin, which is found in your muscles. So microfilaments, these are going to play an important role in muscle contraction. Okay, you've got actin and myosin here in the sarcomere, which as those two shorten and contract will cause your muscles to shorten. Okay, in addition to that, they help with pseudopod movement, the pseudopodium. Okay, so these microfilaments, they're going to help that pseudopodia reach out and grab things so that it can bring in food or it can help the cell to move somewhere. Okay, they also help with what's called cytoplasmic streaming. You see that happening here. Okay, they're helping propel the cytoplasm around here, essentially helping to kind of mix stuff up. Okay, and they will also be found in the microvilli, which are part of your digestive system. Okay, and they help with surface area. They help increase the surface area so you can absorb more. But these microfilaments are really good at bearing tension and helping to resist pulling and stretching forces. Our last kind of fiber that you'll find in the cytoskeleton would be what are called the intermediate filaments. And the intermediate filaments are going to help maintain cell shape, okay, as well as help anchor the various organelles. Okay, but the intermediate filaments are going to be just that. Okay, they're going to be of an intermediate size, kind of in between the tubules and the microfilaments, and their main job would be more anchoring. Okay, forming shape, maintaining structure, and anchoring things. So most of y'all remember that a plant cell is going to have a cell wall. Animal cell will not have a cell wall. Okay, so that plant cell in the cell wall in plants is going to be made of cellulose. In fungi, it's going to be made of chitin. Remember, some of your prokaryotes will have a plant cell wall. Some of your prokaryotes will have a cell wall in general. It can be made out of a variety of things. Again, it can be made out of peptidoglycan. It can be made out of lipids. It can be made out of uric acid. Again, a variety of things. These plant cells, in particular, are going to have what's called a plasmodesmata, and we mentioned that earlier as one of the things an animal cell does not have, but a plant cell does. And you'll see the plasmodesmata are these little channels, these little openings here between the various plant cells, and they allow for cytoplasm to flow back and forth between those two particular plant cells. And so that allows for an increase of communication, increase of flow of nutrients between the various plant cells. So plasmodesmata are a kind of plant cell junction that again allow for cytoplasm to flow between the two cells that they are connecting. Our animal cells are going to be surrounded by what's called an extracellular matrix, which is also known as a glycocalyx. Okay, this extracellular matrix or glycocalyx is going to be made up of glycoproteins. You've got some examples here of your glycoproteins here: collagen, fibronectin. Okay, you'll also see proteoglycans in there. In these various filaments that are there, these various glycoproteins, your collagens, proteoglycans, fibronectins, they're going to anchor into the cell. They're going to connect to the integrins. You see the integrin here, which are the proteins in our cell membrane. Remember, your cell membrane is made up of mostly of phospholipids. Okay, and then there's the proteins embedded within it. So integrins are a type of protein. And you'll see, like here, your fibronectin is connected to the integrin protein. And so they're going to help the cell with adhesion. It's going to offer support. It can help the cell move. They also help. Remember that your glycoproteins that can help identify the cell. They act as like name tags for the cell. Okay, you can see this integrin here anchoring into the cytoskeleton, offering shape and support inside of the cell. Okay, so we've got a variety of proteins that you're going to find in that extracellular matrix surrounding the animal cell. And just like your plant cell, your animal cell will have particular junctions that help hold the cells together. So let's look at a tight junction first. Okay, this one here is a tight junction. The two membranes are touching one another. You can think of it almost like a zipper. These junctions are very important in places where you want to be sure things do not get in between the cells. For example, think your stomach. Your stomach is full, has a very, very low pH because all the acid that is contained in your stomach. You don't want any of that acid to be able to leak between the cells into the rest of your body. And so the cells of your stomach are going to be held together by tight junctions so that they are sealed up nice and tight and nothing can leak out of there. Okay, another one we have here is what's called a desmosome. You can see all these anchoring desmosomes here. These are like rivets. These are going to really anchor and fasten those cells down together. For example, the bottom of your skin cells, those lower skin cells, they're going to be anchored down by desmosomes so that your skin doesn't just easily get ripped off. Okay, so these are anchoring and fastening cell junctions. And then down here, you can see these gap junctions. Okay, and so your gap junctions are going to be very similar to your plasmodesmata in a plant cell. They're going to allow for that cytoplasm to travel back and forth between the animal cells. Okay, so hopefully the majority of this is review. Like I said, very little should be new, but I do know some of the junction things, cytoskeleton stuff, some of that stuff should be new. And it's very, very important, though, that you have got to get this. Some of these things, they just have to be memorized. Okay, you just have to immediately be able to tell me what a mitochondria does and not even think about it. Immediately tell me what a ribosome does and a Golgi body does and the parts what the cytoskeleton does without even thinking about it. So we're going to try our best to do a lot of do some work with these, but you are definitely going to have to just start memorizing on your own as well.